Welcome back, everyone. I would like at this time to introduce Dr. Elaine Hsiao, the Logi Associate Professor of Biological Sciences in the Department of Integrative Biology and Physiology at UCLA, as the moderator of the Moving Microbiomes from Bench to Bedside. Elaine, please take it away.
Great. Thanks very much, Mark. It's a really great pleasure to be able to moderate a panel discussion on the microbiome and specifically its clinical translation and commercialization promise of microbiome-based therapies. I'd like to start by just jumping in to introduce briefly the panelists and actually asking them if they wouldn't mind talking a bit, maybe a few minutes each, of how they made it to the microbiome field. And so let's see. I'll start with who I see on my gallery first. Sarkis Mazmanian.
Thank you, Elaine, and also to all the organizers for including me in this panel. And so just a little bit about me and then how I started working on the microbiome. I actually started my undergraduate and career at UCLA and also continued on as a graduate student at UCLA as well. And so spent 11 years on campus, still feels like home. And then transitioned to Harvard Medical School for my postdoctoral fellowship and then returned to Los Angeles in 2006 to start my own lab at Caltech, and I've been there ever since. Initially, I started working on the microbiome as a postdoctoral fellow at Harvard because having been trained in microbiology at UCLA, I wanted to do something a little bit different than what traditional microbiologists were doing. So at the time, and this is around the turn of the century, most microbiology was focused on infectious disease, micropathogenesis, as well as metabolism. And as I said, I just wanted to do something different. I read a one-page article written by Jeff Gordon about microbes living in the gut of people, largely understudied, a lot of work, that were, decades old at the time. And I just thought it was cool and really jumped in, started working on how the microbiome impacts the immune system in preclinical models, spun out a company around that technology. And we've been working on the gut-brain connection as a lab for about 10 or 11 years now, in mouse models of neurodevelopment and neurogeneration. I spun out a company on that technology as well. And again, happy to be here.
Great. Thanks so much, Sarkis. Thanks so much for participating. We've got next Denise Kelly from Seventure.
Hi, Elaine, and thank you to everyone for the invite, to the organizers for inviting me. So first of all, by way of introduction, I've spent the major part of my career after my PhD, which was in microbiology and immunology, working as an academic with a personal chair in the University of Aberdeen. And I think my journey was a bit like Sarkis. I was very aware of the focus of pathogenesis and host interaction, and really started to ask the question very early, what are commensals doing? And I think I was inspired by Jeff Gordon, because one of the first bugs that I interrogated was a Bacteroides theta-micron, and he was at that time doing the first genome sequencing of that strain. I was really focused on the immune system, just as Sarkis said. So I worked on innate immune pathways such as NF-kappa B, and then really got motivated by the idea and the notion that bacteria could indeed regulate T cells. So with that, I eventually did also what Sarkis did. I set up my own company. I think there was only in 2013, and I think this is incredible, 2013, there were only about 12 microbiome startups, and I'm proud to say that mine was one of it. It actually continues. It's newly branded, differently branded, but continues to grow and develop. So I then left, and I've been with Seventure Partners, which is really why I believe I'm on this panel, and I've been with Seventure Partners for 6 years, and investing specifically in microbiome startups. So thank you very much. Look forward to the panel.
Thanks very much. It's really great to have your perspective here, especially now as an investment advisor. Up next, we've got Nicole Kimes, the CEO of Siolta Therapeutics.
Hi, Elaine. Thank you for the invite. So my path to the microbiome started in environmental sciences. I am a microbial ecologist by training, and I always had two passions. One was environmental science, and the other was human health. And what I came to in my environmental science training was the importance of microbial communities in all different kinds of ecosystems. And it really opened my eyes to the connection with the human microbiome and how that could actually be the interface in how our environment impacts our host physiology. And it was really exciting to make that recognition. So a lot of the tools that we use, statistics, sequencing, all of these really important elements of the human microbiome research that we're all doing was really, much of it was developed in the environmental sciences side of things. And we've known about the importance of these communities for decades. So it's been a wonderful transition over the last decade, seeing this applied to human health. Very exciting and lots of work to do moving forward. So thank you for having me.
Well, all of you guys really alluded to the great growth of the microbiome field over the past decade or so, and especially the expansion of microbiome-related biotech efforts. So I wanted to ask for your input on kind of the trajectory over the past 5 years. What's been driving this great growth? And where do you see the current state of the field, especially relative to other fields that are more well-established? Anyone like to jump in on that? I know I threw in a lot of questions at once.
I guess I could pick this up. I think sort of certainly from an investment perspective. So I've now been with Seventure for 6 years, and they were one of the only funds actually to completely dedicate a fund to investing in the microbiome space. So this was really early. And what we were able to see then were companies, that were pursuing very different approaches. So the kind of journey into the microbiome was quite diverse. So by that, I mean diverse in modalities, diverse in the indications they were targeting, diverse in the approaches they were using. And obviously, in terms of diseases that could be modulated by the microbiome. And really, it started probably with a major focus in immunity, which is not surprising because we know that bugs are regulating the immune system, that's for sure. We've seen that with pathogens. And then it stemmed into metabolic. And then as Sarkis says, we're seeing more and more growth around, for example, gut-brain, gut-skin, and really with that growth in science, so it's science-driven, particularly with HMP projects and MetaHit, science is really advancing here at an incredible rate. I mean, phenomenal. If you look at where we were 5 years and where we are today, it is pretty significant. It's probably unprecedented rate of progress. And with that, we just see companies going from what was 12 in 2013 to several hundred today, with massive, diverse geographic distribution. So each one of them are exciting. And I think actually this is the really right time to hand it to one of the other panelists, because they're each of them leading, one of the front runner microbiome companies.
And maybe I can add some thoughts as well. I fully agree with Denise in terms of really the rapid expansion. I couldn't have predicted this, this many companies and this much enthusiasm around the space a few years ago, but it's very gratifying to see. And there have been advances, and I think the audience either knows and will probably discuss some of the specific advances that have come about in the last few years. But suffice it to say, I still think we have a ways to go, right? It's a new area, relatively speaking, chronologically speaking, and we need some wins. We need some wins in the clinic, and there appear to be some promising signs, a few roadblocks initially, but some promising signs now. But I would say, maybe in response to Elaine's question, that the field is, somewhere in adolescence, right, or early teens, to benchmark against our lifespan, because we're still learning. There's still a lot that we as a community need to figure out, and through the successes and failures, I think we're doing that. And I do believe, and I agree with Denise, that in the next few years we'll see really tremendous successes. I'm hopeful of that. And again, this really relates as everyone would imagine, to advances in the clinic, meaningful outcomes in pivotal clinical trials that put an approved microbiome-based drug on the map, right? And I think that's the stamp of validation for the field.
Yeah, and I'll add briefly to that. We often talk about this from the Gartner hype model for emerging technologies, and you usually have a technology trigger that really gets people excited, and then sort of reality sets in, and it bottoms out at this trough of disillusionment, and then the really hard work starts, right? And then the science kicks in, and he refers to it, or it's referred to as the slope of enlightenment. And I believe we are in that slope. There's a lot of work. We're starting to see some of those successes. We've had multiple phase 3 readouts of late in clinical trials, and additional phase 2 data coming. So I'm optimistic that we are on that road to enlightenment, and we'll be seeing some FDA approvals come out in the near future. If you think about it in the context of sort of cell and gene therapies, there were some early successes years ago, followed by some very dramatic failures. And then of late, you're starting to see more consistencies. Even Scott Gottlieb from formerly of the FDA was stating that he believed that by 2025, they would be giving somewhere around 20 FDA approvals a year in cell and gene therapies. And that's a huge advance over the last 5 to 10 years. So I would say we're not quite that far, but we're hopefully on that road.
Great. And I think more and more, even on the research side, we're seeing that the microbiome is being increasingly implicated in a wide variety of disorders. And so where do you see the current landscape of using microbiome-based therapies? And where do you also see the biggest opportunities for continued development?
I think it's hard to argue that a lot of the most fundamental research to date has been done in fecal transplants. I've been using fecal transplants as a modality, and with an indication specific to Clostridium difficile infections, which causes pseudomembranous colitis. And there's precedence for this, right? So there's just a number of clinical studies over the years, over the decades, actually, using fecal transplants for C. diff infections. And so a number of biotech companies have that as a large focus of their programs. And so I think it's a starting point, right? But in many ways, I think that the extrapolation from C. diff infections to other disorders isn't a trivial one, or isn't an obvious one. C. diff infections are largely, I like to think of it as like the FMT is putting out a fire, if you will, right? We're not asking much of the microbiome compared to other indications where the microbiome is tuning the immune system, or tuning the metabolic system, or the nervous system, or affecting its functions in ways that the microbiome, we're asking the microbiome for those, for additional indications to actually do more than just microbe interactions. We're asking for micro-post interactions. And so, but that being said, I think, as was already stated, thinking about inflammatory disorders, allergic autoimmune disorders makes sense because of the intimate relationship between microbes and the immune system. It's a little bit of a twist on what we've classically thought in terms of immune function. In this particular case, the immune system isn't controlling or limiting an infection. It's responding to microbial colonization and potentially benefiting from it. And if we can harness those processes and harness those technologies, I think maybe for, conservatively speaking, maybe Crohn's disease, maybe an indication where we may see successes in the near term beyond C. diff infections. If I were to place a bet, I think the first drug will probably be in the C. diff space, just based on history and based on the amount of activity, but potentially, again, inflammatory diseases following.
Yeah, I think just to add to that, I think, there's no doubt, the FMT, we see Rebiotics, Ceres and Finch coming through, and certainly, clear hints of BLA approvals coming. I think on the immune side, where we see a lot of activity and a lot of clinical trials, and actually quite fast-track clinical trials, is obviously in the immune oncology space. And I think there was a huge interest in how the microbiome is modulating the effect to checkpoint inhibitor. And, we've seen a lot of the companies turn their pipeline to really exploit that. And I think some of the potentially early wins could come from there. That's a sense. But I do agree with Sarkis, getting an FMT approval through phase 3 is one thing, but I think we really will see a massive surge once we get through the treatment of the more chronic diseases, the more complex like IBD and cancer.
Yeah, I would agree that the future is definitely, there's a focus on systemic impacts of the gut microbiome and diseases that have really chronic and complex pathologies. And I think one of the things we have to keep in mind is not just microbiome specific, but really with an overall focus on moving towards precision medicine and really having more targeted therapies. This is an area where the microbiome, being an emerging field, has the opportunity to do it from the outset and really to integrate some of those concepts as we move forward with whether it be targeted diagnostics or different formulations for different portions of the population. It's a really exciting area where we can meld this new technology with a different approach to disease.
And I would agree and just highlight this point of personalized or precision medicine, right? So the microbiome, each of us have our own microbial fingerprints, so similar to our genomes. So it's different when you think about drug development. It's not going to be maybe one target or one drug for everyone, but potentially a match of a particular microbiome and its function to a person's genetic landscape. And so, yeah, I definitely echo this point of really thinking about drug development from a personalized approach lens.
And on that note, I mean, you all alluded to how these early wins may be in the FMT space. What are the implications for other treatment modalities, especially as you mentioned, precision medicine? Are we envisioning mostly probiotic or live microbial administration, or are other modalities potentially more promising?
I have to just jump in there. I couldn't stop myself. So this is the big debate, isn't it? It's, I mean, there's so many different modalities. So you have, groups that are pursuing single strain. And the number of times I'm asked the question, would you bet on a single strain or a consortia? Obviously pharma, they like the small molecule, and we've got companies like Enterome, which is in our own portfolio, our second genome, really trying to dissect peptides, molecules from bugs that are bioactive, and they clearly are. I mean, the science is there to support all of this. I think the big win, I personally feel, I have a slight sort of view now, just with the FMT data and looking at even what's happened with COVID, understanding that a bug doesn't exist in isolation, that there's a huge collaboration interaction, and it is an ecology problem. I still believe, that we will see some wins from a more consortia where you're exploiting pleiotropic mechanisms, and exactly to the point. I think it's Sarkis and Nicole said that, Nick said that, these are really complex diseases, and so it really is. They are perfect for a consortium microbiome intervention. And I guess what we're able to do now with just technology is to really understand mechanistically how these products are collaborating to bring resolution of disease. So I think that's a key point, for me. So it just continues. I think the area of personalized, as we touched on, I think is a big data issue, which I'd love to hear everyone's view on. But when you really get to disease and you've decided to go down live biotherapeutic, live dead bugs, you really are tapping into disease mechanisms and how those organisms that you've selected for your consortia really plug all the problems in the disease, the different disease targets.
Denise, I would add to that, if everybody can hear me now.
Yes.
Yeah, so I'm the unfortunate beta tester for Elon Musk's new Starlink internet, so I apologize for that. It's clearly not working as well as he claims. Denise, I really, obviously, I've I've spoken with everybody here individually and as a group ad nauseum about this ecological approach and consideration. And Denise, I would even take it one step further, that you have to modulate the native environment for the ecology that's being transplanted for it to be efficacious. So does it need to be in the mucosa? Does it need to have a certain pH? Do there need to be other metabolites or a certain immune response in the host that makes even the transplantation or the modulation of that ecology effective? And so I think that we're just scratching the surface of the extent that the ecology drives the microbial ecology, host, microbe host, and the way they interact together. So whether it's a small molecule, which may be immune to some of these ecological considerations, or whether it's a single strain or it's an entire transplanted ecology, I think we need to consider the environment within the host as well.
I think part of the answer here is also, again, our ignorance about the impact of the microbiome on various different biological functions and organ systems, right? And so there are examples of particular organisms, particular pathways, whether they're small molecules or other products of the microbiome, that seem to have effects of interest, essentially working like a drug. But the fact of the matter is we still don't know a lot of what the microbiome is capable of. And so it's a continuum or a spectrum, right? And so you can start with fecal transplants, which presumably have a multitude, or we know have a multitude of different activities. And so they may or may not work in certain people, but you're covering your bases without fully understanding mechanisms of action. But as we research more, there are going to be, again, more refined therapeutics based on more refined biological connections to the host, again, the immune, metabolic, or nervous systems, or other systems as well. And so I think as we learn, we can get to more pharma-friendly modalities, small molecules, and more protectable, if you will, if you think about regulatory or IP considerations, drugs as well. But in the meantime, I think, again, the workhorse for the foreseeable future will probably be FMTs. But as more and more modalities come out, I think over time we'll probably see, my prediction is we'll probably see a transition towards more of these refined molecules or modalities like biologics with more known about their mechanisms of action. And I think a lot of the companies, right, that are developing FMTs are also starting to work on exactly how their FMT performs the functions that they're most interested in.
Just to be provocative, I do want to jump in on the point about personalization, because look, most of our investors are technology investors, and I hear personalization so often, and in so many ways I agree with it. But the fact of the matter is that FMT, taking stool from potentially a random person on the street that donated to a bank, is efficacious for some infections across an entire heterogeneous human population. So we know that at the mammalian level, and even seeing translation of efficacy from mice models into humans is in some ways the antithesis of personalization. It's showing that it's not even just at the human level, but at the mammalian level, some of these features get conserved. And so I think that discussion gets lost or overshadowed in the hype and the future orientation around how sexy personalization sounds. Now, there will always be responders and non-responders, which I think personalization is just a proxy for discussing, is that there are going to be some people, and we've seen that in checkpoint inhibitors and cancer immune host microbe interactions more than microbe interactions. But it does remain true, and it needs, the personalization discussion and hype does need to be balanced with the fact that we do most of our discovery work in mammals that are non-human, and we do expect to have a conservation at the human level. And FMT is a perfect example of healthy versus non-healthy, or ecology versus ecological disruption being a very binary outcome within a human as an intervention.
Yeah, I think that's a great point, Raja. And I think one of the things on the FMT, we're seeing a lot of the companies, a lot of the publications really addressing the responder, non-responder, because you can interrogate the data, looking at the engraftment, what is engrafting, and trying to link that back, that signature, microbiome signature to response. I think that's a great way, but I'd love to turn it a little bit as well, because that's a great way. You're starting humans, this is kind of the type of strategy we see with a lot of companies at FMT, like Matt Pharma, like Finch, human first, and then interrogate, and then build towards a product. But I'm really curious about phenotypic screening at the model level, either in vitro or mouse models. And we know the mouse is the workhorse for drug discovery, and we use it in the microbiome. So I'd love to hear a little bit of debate, and I sense I'd love to push this towards both Nick and Sarkis and Raja. Just what do you think? Do you think these are valuable models to get us through the preclinical validation to convince, FDA that this is something we can bring to human? And how useful do you think it is, and how risky do you think it is?
Well, I think to your point, it's in our minds, it is absolutely imperative that we start with human data, and that we're starting from really clinical indications of what is distinguishing between health and disease. And that sort of speaks to Raja's point of there are generalizations that can be made, and I don't mean to discount those in any way, shape, or form. But then moving into the screening and validation and mechanism of action, I think you have to use lots of different tools to answer different questions and make sure that you're looking at different aspects. Do I believe there's a single tool out there that is the take home, and if you can show efficacy using that tool, or an immune response using that tool, that magically will translate? I think that's too simplistic of an approach. But there are lots of different tools that can at least help us understand the mechanism of actions to really de-risk some of the applications that we're all going after. I would always emphasize using lots of them.
I think the microbiome provides a unique opportunity, as Denise alluded to go direct, to take drugs directly into humans, right? And this is challenging for other drug development campaigns and other areas, right? Because if you're looking at novel molecules, novel chemical matter, it's difficult or impossible to do your first trials in humans. So you do rely on mouse models, which are imperfect, which don't, capture diversity of the human population, which obviously have many shortcomings as well. But again, I think the microbiome, because the drugs come from humans, and the FDA realizes this, and the FDA is bullish on taking organisms from humans and putting them into humans, it does afford us the opportunity to collect a lot of clinical data. Maybe it's not even interventional data at first, right? But a lot of clinical data very early in a drug development, in the life of a drug development program. And then I very much agree with Nicole, then you have to use additional tools, right? Whether they're, animal models or computational tools and other technologies to really round out your understanding of what's happening, even if you have a clinical success. But again, that's one thing that I think is relatively unique to this field, is the ability to very quickly translate early research findings into clinical efforts.
Great.
I guess along those lines, and I guess in the spirit of the title of this panel, what body of research is needed to enable translation and commercialization? I think that, some of the promise of the microbiome is that there's all this unknown functionality that's in there, and it's almost like a treasure trove of new biology to understand. And, for somebody starting out from the beginning, what body of work is really needed to enable translation? And also, what are the current challenges in translating basic microbiome research to clinical application?
I'll start. I think it depends on which microbiome you're targeting. If it's the gut microbiome, then obviously the human gastrointestinal tract is incredibly unique. If it's the vaginal microbiome, it's the same case. Humans are the only mammals that are dominated by vaginal lactobacilli, so you'll just have a very hard time translating that in anything outside of a human model. I think there's an underutilization of the engineering problems about microbe delivery, getting it where it needs to go. Does it need to be a mucosal organism? Does it need to have a luminal delivery? Does it need to have colonization? Does it work through engaging with immune cells? And if so, you can have the perfect consortia, but if it's releasing too far down in the small intestines, you'll miss your target. So I think that the biology and the translation can't be over-reliant on the biology. And I think that partially explains why some animal models, which, by the way, I think humanized animal models are incredibly useful to answer gastrointestinal research questions related to microbe, and even microbe-host interactions. So I think it's so dependent on what you're targeting. And I would encourage close collaboration with some of the technological and engineering and typical, even just drug delivery system models that help you understand what's going to happen. We oftentimes use an external gut that mimics the human gastrointestinal tract, and I've found that data can rapidly iterate on the release profile that we're looking for to deliver an organism gastrointestinally where we want. And so my fear is that the perfect biology can get overshadowed if some of these other considerations aren't taken into upfront, and creates a lot of false starts for companies that have good biology, but maybe didn't think about some of the engineering or technology hurdles.
Yeah, I think from probably an investor point of view, you think about diligence, what you want to see in a package that you think, right, this is kind of bench moving forward really into development, right, to commercialization. So you're seeing the whole thing as a product. You know, if we simplify it as a product, it's got to be a new drug. For us, particularly in the microbiome space, a lot of the signatures, and you've heard all of the panelists, come from human first data. And if you think about that, the workflow and complexity of producing that data and really believing in the robustness and absolutely not even believing, having the standards and standardization that demonstrates that it's really robust data, I think is critical here. So things like, having sort of well-defined either cohort numbers, pretty large actually. It seems now we're talking about, to get a sort of meaningful signal, you certainly need to be in the hundreds. And if we believe our colleagues in the Weizmann, it could even be thousands. You've really got to think robust data, strong analytics, machine learning, and I think that is a key part of this field, so that when you start to advance your product, you really are starting with, really underpinned science and the reason to believe and to go forward, regardless of all the screening you do thereafter. So I think there's a lot of groundwork around sequencing and understanding what a microbiome is, what's there, how it's changing over time longitudinally, rather than just this one-shot analysis. All of these things, I think there's a much more heightened awareness of the rigor by which we have to approach these type of science now.
And I think this point connects to the personalized or the precision medicine topic as well, right? Because when you do these large studies, you can find subpopulations of individuals who either respond differently or have a different profile that affects the data. And we see this in a lot of clinical trials, right? But I think one of the advantages of the microbiome is you may be able to actually do something about it, right? So if it's a lifestyle or, again, a genetic predisposition that subcategorizes a person, let's say responder, non-responder to a drug, it's more challenging to overcome that hurdle than it is to manipulate the microbiome. For example, if there's a person who's a non-responder, like the IO example that Denise gave, right? So there's a number of ongoing clinical trials now that for checkpoint inhibitors, the people who don't respond may become responders if you give the drug plus a particular probiotic. So again, another advantage, I think, of this field is that, as we learn more about how the microbiome affects, the diversity of the human population, we may be able to very quickly translate that into changing outcomes in the clinic.
I want to pull up one of the audience questions. Lakshmi Reddy asks or comments that there are controversial, though not conclusive, reports of microbes in other tissues like blood and brain in cases without inflammation, and would like to know your opinion on these. And so to broaden this scope, I guess I'd like to also welcome you to just address about any, treatment modalities of targeting microbes to particular tissues. I think we're seeing more of this, and particularly with in oncology, and any concerns or challenges, particularly with that.
I'll start just because I love this question a lot. Actually, it's a big research question that I've invested a significant amount of my own time and resources exploring. There was a big Science and Nature publication that was pretty radical at the time that came out about 2 years ago saying there was a brain microbiome. It was a group that saw microbes in brain tissue that came out of the University of Alabama. And again, I got a ton of pickup and started to sensationalize a little bit for other reasons than just the science itself, because it was a very provocative point. So much so that we started a multi-year collaboration with a researcher at the NIH that did fresh brain tissue over dozens of samples to actually look with the right screening techniques and cleaning that data to crowd out the human genetic information to see if we could pick up a microbial or even a viral signature. And we got all of that data back just a month ago, and we're working on a publication right now. In confidence, just to share with the group, we, with the NIH, did not find any bacteria in healthy or diseased brain tissue itself when collected from a live host. So I think that there's a reproducibility issue. I think that there's a bias in science that if you can publish something provocative first and be the first person there, whether it's reproduced or not, it's an issue. I do think, however, to balance that out, there are microbes in other tissues that are profoundly important to understanding human biology and also engagement between the microbiome and the immune system. For example, I believe that Fusobacterium is not only found within tumors in colon cancer, but is a central part of the driving process of carcinogenesis in colon cancer. And that is microbial translocation within other tissues. I think we've seen that in other organ systems. I just can't understand at the rate at which microbes evolve, why the human brain would be permissive of organisms, because the potential for a microbe to evolve that has opportunism or pathogenicity that takes such a sensitive tissue far outweighs, in my opinion, the benefits that allowing that colonization could confer. So I don't think we know the answer to it, but I am very skeptical on whether there is a brain microbiome to the question's specific example. But I do think that there are microbes in other tissues that are important, both in commensal protection and coexistence within that organ system, as well as driving carcinogenesis or driving some dysregulation.
Maybe three quick points on this topic. First is just to orient the audience. Oftentimes we associate identifying microbial DNA or signatures of microbial DNA as there being bacteria, and they're not, right? And so you can, there's actually a lot of evidence now about cell-free DNA and RNA of various different origins, human and microbial, in the blood. So again, a distinction between live organisms and their genetic signatures. That's number one. Number two, I agree with Raja that there's been a lot of hype around the brain microbiome, but we also, in an NIH-funded project, looked in 500 Alzheimer's brains using sequencing. None were positive for any microbial reads. And so again, no one study would be conclusive, but 0 out of 500 using the most sensitive techniques in our study. And then the other point I'd like to make is beyond just where bacteria are or can be, again, and we're learning a lot about that, is, an organism may not be in a particular tissue to affect that tissue, right? So organisms, microbes make molecules, small molecules that bathe all of our organs, including our brains. There are hundreds of microbial metabolites we know in the human and animal brain, right? And so microbes can have their effect on, again, any tissue in the body without actually being there.
Yeah, I would add to that, to Raja's point of different tissues having different impacts from microbes. The lungs for a long time were thought to be sterile. And that's an area where there's a lot of work that's been done to establish the fact that there are microbes present in lung tissue. So I think we will, I think the body of knowledge and work to really tease apart these questions is starting to occur. And it's important that we do continue to push those boundaries. Because as most of us said in our early introduction to the microbiome space, we typically focused on pathogens, and it's really only been in the last decade that we're looking at the whole realm of the microbial community. So I think we will be changing some of our assumptions as we move forward as that body of work grows.
Yeah, and also I think, sort of the reports, I mean, you probably also see in fact, I was part of the Hanson Wade maternal infant microbiome conference, which is running now. And there's a debate again about, infants in utero and exposure to bacteria. And again, you can see papers that are coming out from groups where the data looks robust. Obviously, the point being, these are really low biomass issues. And I think, the standards in science that are needed to completely validate are really high. So I think I would be very cautious, but I also think, just as Nick said, we need to push the boundaries because, maybe there is some sort of trophism that's important that we're not seeing. We don't know why. And certainly if you look at some of the work from Rob Knight, clearly he's now reporting, cancer-specific type microbiomes that are found in tumours. And so, these are in disease settings. So perhaps, certain diseases, leaky gut, whatever the mechanisms, we don't know. But certainly, I think it's something we still have to kind of push on. And eventually, hopefully, we get to a sort of scientific consensus on whether it's real or not real and whether it's biologically relevant or not.
Great, thank you. And I can't believe it. We're already close to time. I'd like to just end off now with one overarching question for each of you of just what would you like to see happen in the next 10 years, 20 years in this area, whether it be in, basic research and discovery in the microbiome field, in regulatory or commercialization or clinical applications? What would kind of be, a dream outcome for you?
I'm going to steal the first one. For me, I really want to see, I'm a passionate, absolutely, microbiome and the ability of bugs to steer biology is profound. And for me, to see an approved product, cure, literally bring, so if we look at response to checkpoint inhibitor, 30%, if we could get that up to 60, we have made an impact. And that would be, for me, the most wonderful outcome. So, and I hope to see that in the next 5 years. It would be phenomenal.
I have a whole list. I will try and keep it succinct. I think technically seeing standardization so that we can actually benefit from each other's data across the board would be a great advancement to the field in general. I think from a regulatory perspective, seeing not only a approval for a treatment, but I also think we have a unique opportunity for prevention here. And for some of these chronic diseases, could we actually prevent it from ever occurring? I think that would be a huge leap forward for medicine in general.
And I'll obviously agree with both Nicole and Denise. We want to see, treatments. We want to see improvements in human health. So that would be, certainly high on my list as well. But one thing I would add is, just over the years, I've just found that the lay observer of the microbiome, the non-specialist, the non-scientist, both understand the microbiome and are really enthusiastic or intrigued by it. And so maybe the microbiome is an avenue to gain more acceptance among society or for science and the scientific process, or also make people more aware of their bodies and aware of how their digestion could affect other aspects of their lives. And so maybe there's a trickle effect outside of science and development and innovation, but more towards society using the microbiome as a vehicle to understand more about their own health.
And I'd like to see more of a focus on these critical windows of development. So when an infant's born, the microbiome that develops over the first 2 years of life is what the immune system and most of the hosts is comfortable with over time. And so my hypothesis is that if there is a focus on priming that infant microbiome in ways that develop it to be optimal based on what we know and will continue to know, and then perhaps bank that at the age of 2, 3, or 4, and then in periods of duress over that person's life, you can re-inoculate them with their own entire self-FMT from a period of windows of development. I'd be very curious to see. I know that's a big operational and logistical problem, and stool banking itself already with donors is hard enough, but I'd be so curious to see if that could have applications all the way from after a course of antibiotics as a form of self-generated probiotics in the future, all the way to preventing things like neurodegeneration at the other end of the life spectrum.
Great. Well, it looks like we've all got a lot to look forward to, and it's a really exciting time to be in this field. Thank you all so much for participating in this panel.
Yes, thank you, everyone. And thanks, Elaine, for
Thank you,
moderating.
everyone.
Thanks.
Thank you.